All right. Okay, everyone. This is yet another episode of our Western Biology Evolution Seminar Series, or WBESS for short. And in fact, this is already our 12th episode. And today I have here with me Dr. Alberto Perez-Posada. He is a postdoctoral fellow at the University of Exeter. And today he'll be presenting to us a little bit of his work that he did during his PhD on the evolution of hemichordate expression dynamics. So, Alberto, it is a pleasure to have you here with me today, and take it away.
Thank you, Mateus. It's a very nice opportunity, and I'm very thankful for this because I was just telling him that people in our stage, we really appreciate having the chance to showcase our research and what we've been doing. Just as he said, my name is Alberto. I am a postdoctoral fellow. I am currently in University of Exeter at the Living System Institute. I am in the Stem Cell Biology and Evolution Lab with Dr. Jordi Solana as my PI. And today I'm going to talk about some of the work that I've been doing in my previous postdoc. And for that, it's just as he said, like the title, hemichordate cis-regulatory genomics and the gene expression dynamics of deuterostomes. And for this, I'm going to go very broad at the beginning. I'm going to start with one cell because that is just that, as we know, is cells are the fundamental units of eukaryotic life, and as we all know as well, they harbor the genetic material that is required to do basically everything that they need to do and that needs to happen. That's a lot of responsibility, and there are different things that are very important for cells and that they need to happen in the right way, in the right order, and they should not interfere, right? One of them is, for example, the ability to generate new cells, replicate the genetic material, and ensure that everything is split correctly. And this needs to happen in a very controlled way, which is why there is a tight regulation of proliferation for cells, right? Let's call this process A. There is another important thing that needs to happen, and or that cells can happen, which is that when they need to turn into something else, let's call like a cell state, or like when they need to become a different cell type in the body of a larger organism, right? Let's call this process B, differentiation. And these two processes can collide or they can interact together or not, right? But the thing is that one of the layers that the cell has available to regulate all of these is gene regulation, right? In the form of what we call gene regulatory networks. These are groups of transcription factors, genes that control the expression of other genes, and then they are able to bind to the genome, and they act up there and they do their little things, and they are capable of basically telling the cell in a molecular way what needs to happen, right? And as I was saying, there's these two processes, which is controlling when the cell needs to divide or when the cell needs to become something else, that they can be either interrelated, they can exclude each other, and so on, right? But in the end, it's this interaction between the two what allows what we call animal development, as we understand it in a very broad sense. For example, we can understand animal development as the embryogenesis, the process of going from one single cell to becoming a whole organism. And this is the result of the interplay between proliferation and differentiation. This is also so this is development, but development can also, we can understand it as, the process of, the turnover of the different cells, replenishing them when they are wrong and you need new ones. And this is something that we do throughout our whole lives. But also another developmental context can be also regeneration, when some animals, amazingly, they are capable of regenerating whole structures, right? If they are lost or if they are wounded, etc. So why am I telling you this? This is basically one of my biggest questions and my biggest passions in biology. Is one of the things that I am the most interested at. And I am very lucky to have been able to dip my toes a little bit into many of these different processes throughout my career. I began doing my PhD in the middle of 2016 in the Institute of Evolutionary Biology in Barcelona, under the wing of Iñaki Ruiz Trillo, an amazing PI. I had a lot of fun there, and we were studying the origins of animal multicellularity. In that time, it was from a genomic and functional genomic point of view, and we were, putting a lot of effort into turning these unicellular relatives of animals into experimental organisms via, making genetic tools and understanding them a little bit better. And I was working specifically in the cell cycle of one of these unicellular relatives of animals, and this put me into contact with the world of cell proliferation, which I love. Then after that, I moved on to work on the evolution of genome regulation during development, and this was in the Andalusian Center of Developmental Biology under the wing of Professor Jose Luis Gomez Carmeta, who sadly passed away during that time, but also with Juan Tena, who was like an amazing mentor. And actually, the work that I'm going to talk about today is from this stage. And then afterwards, I moved to the UK and to the stem cell biology and evolution lab with Jordi Solana, first at Oxford Brookes University and then at University of Exeter. And here, thanks to what I learned during my previous stages, I realized that it would be amazing to study all of this with a much deeper level of resolution. And for that, I went on to learn single-cell transcriptomics and basically genome regulation during stem cell differentiation. So this has been my career so far. And as I was saying before, like the work we're going to talk about today is from the first postdoc, right? And in order to transition into that, let me get back to animals and how many they are and how different they are. This is animal diversity as we understand it. And as you can see, they form like a very nice group with their unicellular relatives. And we have these early branching animals like sponges, ctenophores, cnidarians, brachiosaurs. And then we have this group that I have highlighted here that is called Bilativia, that it's basically what most of us are most familiar with because the largest diversity of body plans and life strategies that we have seen in animal life stems from here. And this is where basically we are, like as vertebrates. Specifically, we are part of deuterostomes, this second group that I have highlighted here, that I have more in detail here. So as I was saying, bilaterians, which are animals that have bilateral symmetry, basically their bodies can be split into symmetrical halves for the most part. They can be divided into protostomes or deuterostomes, where we are as vertebrates and as chordates. Deuterostomes are a very interesting group because they share a number of developmental characteristics. Like, for example, what gives their name is like the first structure that forms in the embryo is the anus, not the mouth. And then they also share, like, for example, pharyngeal gill slits, which are the openings like for the gills. During development, at some point, they have them. They also have, for example, they form the skeletal structures from the mesoderm. They have enterocele, which is basically the coelom is formed from the endoderm. And all of these characteristics plus molecular data, like, for example, phylogenomics and so on, they position them as, as a relatively consistent group. And for me, what's amazing is that the diversity of body plans, specifically because we have a group here that is called the echinoderms, that they have acquired pentaradial symmetry secondarily during evolution, right? And this makes a bit of a puzzling scenario because these kind of comparative approaches that have been classically performed can be done, but I don't know, it's like the echinoderms are there, we are not pentaradial, so like how do you match this a little bit, right? But luckily for us, we have this group called hemichordates, I have highlighted here. I don't have the name, I'm very sorry. They are these little worms on the left of echinoderms, that they are a little bit of in between of what we understand as chordates and echinoderms. Why? Well, for one, they form a group with echinoderms called ambulacraria, and that is very interesting. Molecularly speaking, they share more similarities at the gene content and so on. But still, like hemichordates, they retain their bilateral symmetry, even in adulthood, which is something that echinoderms don't. On top of that, hemichordates, they, not all of them, but many of them, they showcase indirect development. Basically, they have, after gastrulation, after let's say the canonical embryonic development, they develop into different body plans and body shapes that they are different from the one that end up being as an adult, right? And for example, they swim freely in the water column, which is the same that echinoderm larvae do. They share certain structure similarities with larvae from echinoderms, but yet when they develop as adults, they go back to they go to the sun and they turn into these benthic animals that they burrow in the sun, and they have this bilateral symmetry. So comparing these different groups of organisms within deuterostomes can inform us about what we call the last common ancestors, different ones. These are the organisms that we can imagine that they lived at the time of the splitting in this tree of life, right? For example, if we compare chordates to echinoderms and hemichordates, we can learn a bit more about the last common ancestor of chordates, but also the last common ancestor of deuterostomes, And if we compare deuterostomes to more protostomes, we can also understand a little bit better the last common ancestor of bilaterians. So talking about hemichordates, which is what we're here for today, as I was saying, they are these bilateral organisms, this group of benthic borrower worm-like beings that they live in the sea, and they are tiny and inconspicuous. They do their little thing, and that's it. Sometimes they form these little colonies that you can see, like in the picture down in the left, and they can feed from things that are swimming in the water column, and that's it for the most part, you could think. But interestingly, as I was saying, they have this bilateral symmetry. They have this anterior-posterior patterning that is reminiscing of some things that we see in chordates as well. For example, like they have three clearly different places, like the proboscis, the collar, and the trunk. And the resemblances are not only at the morphological level, but it seems that some of this is also at the developmental level. So the thing is that precisely because hemichordates have retained the bilateral symmetry, they are more easily or more straightforwardly comparable with chordates and other species like other protostomes. And from this is how we have learned that some of these mechanisms, like for example, the process of what we call neurulation, the formation of a neural tube, which they also have, by the way, which is also similar to chordates. And it occurs in a developmentally relatively similar manner to what happens in chordates. And also these resemblances are not only at the level of, this germ layer or these territories and these processes that are happening with the cells during embryogenesis, but also like the molecular signature. And by molecular, I mean, for example, the expression of genes that are important to specify the patterning of the, in this case, for example, the anteroposterior of the organism during the larvae, but it can also, these mechanisms resemble, or these molecular signatures resembles those of the structurization of the central nervous system in chordates. So this is really interesting because certainly it suggests that some of these mechanisms, they might date back to the origin of chordates, and they might be even older than we expected or we thought. Until now, there have been some efforts into understanding a little bit better if these similarities go beyond simply gene expression, and if the extra layer of genome regulation, as I was talking about before, of gene regulatory networks, if this is also certain more or less similar, right? There has been some efforts into assessing this, especially in this species called Saccoglossus, which is a direct developing hemichordate. But so far, there has been, until now, a lack of high-throughput, genome-wide, quantitative, genomic and transcriptomic data. We know since the advent of genomic technologies that these kind of big data approaches can effectively generate or formulate many new hypotheses and can give us a lot of new perspectives into these questions. And in the case of hemichordates, until now, sadly, we didn't have much. So, of course, like there were genome papers before in the past, and they have been really informative, but all of these additional layers of how genes are expressed, how is the chromatin organized throughout the genome during development and during the temporal life cycle of this organism, we haven't had it. But so this was a little bit the mission when we began. And luckily for us, this turned into two incredible, amazing works that I have been very happy to work in collaboration with other people. For which we have this new genome assembly of hemichordates, and especially what I'm going to talk about today is the first publication that I'm showcasing here, which is the one that I participated the most. That was published very recently, where we assess a little bit what is going on at the gene expression level and at the chromatin level in hemichordates. So let me introduce you the main character of this story. So this hemichordate is Ticodera flava. It's one of the indirect developing hemichordates. They undergo through a life cycle that has different larvae, as we will see in a minute. And in this case, for this project, these were collected in the Penghu Islands in Taiwan by our dear collaborators, the Yishen and Sky Yu Lab in Taiwan. And as I was saying before, similar to other hemichordates, they are very inconspicuous. They just burrow in the sand, but they undergo through this amazing biphasic, what we call it, a life cycle, which means basically that they undergo what we understand as, early embryogenesis. And at the moment of gastrulation, they turn into these different larval stages that they swim freely in the water column until they transform into an atoll. right? So the idea here was trying to characterize at the molecular level as many life stages as we could. And this first at the gene expression level, and then we decided to delve a little bit into the chromatin regulation level. And for this, we generated 16 different transcriptomes of different life stages that I'm going to talk about a little bit now. So the first thing that we did was, of course, we generated the transcriptomes, we sequenced them, mapped them to the genome, and we were able to see that they resemble more or less what we were imagining, which is basically that we have this biphasic life cycle that we can see at the morphological level, and this translates into some molecular, features. What we see here, for example, is that larval transcriptomes, they tend to group together and correlate more highly compared to, in relation to the other transcriptomes, and the early embryogenesis transcriptomes, they tend to group together. They tend to be more similar. And gastrulation is a little bit in the middle. It seems to correlate both with early embryogenesis, but also with post-embryogenic, let's say, indirect development transcriptomes. And in order to understand a little bit better what's going on and what are these genes really, the ones that are similar, and how these genes are being expressed throughout time, what we did was looking for expression patterns throughout development. We did differential gene expression throughout development of this species, and we retrieved groups of genes that behave more or less in the same way, what we call temporal cluster of gene expression. We did about 22 of them. This accounts for nearly 80% of the whole gene content of the organism, but we shouldn't be surprised by this because we're talking about, gene expression throughout the whole life of an organism. And on top of this, we were thinking, right, gene expression tends to be one of the biggest components of gene expression is regulation by transcription factors. So we were wondering if there could be any kind of structure into the data that are basically transcription factors. For this, we classified all of the genes that look like transcription factors in Ticodera in different classes based on their similar sequence homology and so on. And we were able to interrogate whether transcription factor class abundance throughout the development changes at any point. And by doing this, we were able to see that certain classes of transcription factors, they tend to be transcriptionally more abundant during certain moments in development than others. And this is reassuring and nice because we can see, for example, here the PO and E2F classes, PO, which is one of these big initiation factors that have been described for the beginning of development. But E2F is also a regulator of cell cycle and cell proliferation, which is nice. And then we also have the contrary, the opposite case, which is some classes that are more expressed later in development. For example, the beta zip, the basic helix loop helix, and for example, the steroidogenic, like transcription factor class, which is the one that is here in purple at the second half of this heat map. That this one is related in many cases to the control of gene expression involved in production of hormones, that some hormones are involved in metamorphosis in several cases for several species. So that is nice, that is reassuring. So the next thing that we did is, we did a little bit of a similar analysis, which was basically correlating these classes of transcription factors with the different temporal clusters, and this gave us a little bit of the same picture with a little bit of more resolution. And so far, so good. This allows us to say that, or see that hemichordate development follows a molecular biphasic program controlled likely by different classes of transcription factors. But the bigger question remains there. What is the whole picture in relation to deuterostomes? As I was saying before, the cool thing about this is comparing these different species and trying to assess what are the similarities and differences, right? So for that, what we did was we relied on data that had been publicly generated before, available before, from echinoderms and cephalochordates to try to understand a little bit better if these organisms are more or less similar during development, right? So the first thing that we did was we generated orthology relationships for all of the genes between the different organisms that are of importance in this study. And this is, for example, comparing hemichordates with echinoderms. What we saw here, what we did was basically correlating the different transcriptional stages of every organism between each other using these one-to-one orthologs as a translation layer between the two species. And this allowed us to see which pairs of developmental stages between pairs of species were more similar. And this is what allowed us to see, for example, in the case of echinoderms, that the most similar stages with hemichordates tend to be late development. There's also certain resemblance between early development in both species. And in this case, early development seems to be enriching neurogenesis and germ layer fate commitment. This is very nice. We did this for both for the purple sea urchin and for Enicia japonica, which is an early branching echinoderm, which allows us, to have a better comparison there. And this is where we saw these similarities. And then when we compare to cephalochordates, which are chordates, as we saw before, we see that gastrulation is more similar to neurula, which is not exactly the same as gastrulation in chordates. It's a developmental stage that occurs slightly after. And what we saw here is that these similarities are enriched in neuromigration, cell fate, and anteroposterior patterning processes, which it already paints a different picture between what makes hemichordate gastrulation similar to echinoderms and what makes hemichordate gastrulation similar to whichever stage in cephalochordates. But the thing when doing this comparison is that we were using one-to-one orthologs between set independent pairs of species, right? So we cannot ascertain with a lot of certainty whether one stage of hemichordates is more similar to one or another stage of different species. So for doing that, thankfully, we relied on this other publication that I said before, that was published in PLOS Biology in the very same year, where we assembled, there was a chromosome level assembly of hemichordates that was compared to the genomes of other species. And this allowed to detect ancestral linkage groups, whether we can think of them as syntenies. And by doing this, we were able to elucidate like the relationship, the orthologous relationships between a large number of orthologs of genes between three species, purple sea urchin and Branchiostoma floridae, one of the cephalochordates. And this, we reasoned that this is the toolkit of genes that we were confident that they were present in the last common ancestor of deuterostomes. And this allows for one-to-one comparisons between the species, right? So when we did this and we repeated our analysis, we saw that gastrulation of hemichordates is likely more similar to the cephalochordate neurulation than to echinoderm gastrulation, which means that it suggests that this toolkit of genes from the last common ancestor of deuterostomes has retained their molecular dynamics, their temporal dynamics during development in hemichordates and cephalochordates, and not in echinoderms. But on top of this, going back to the transcription factors, because we always go back to the transcription factors, we were thinking of which of these genes that we assess as one-to-one might be regulating gene expression of the rest of the genes. So basically, we subsetted and identified these groups of transcription factors that showcase similar expression patterns across the three species, right? And for example, I want to focus here in one of these little groups of transcription factors that they showcase a similar expression pattern. And this is what we can see here, which is basically a group of genes, including guscoid, FOXA, brachyury, NKX transcription factors that are well known, that they seem to be expressed during mid-development in ambulacraria and slightly later in cephalochordates. So this suggests that processes occurring after chordate gastrulation could have been deployed a little bit earlier in ambulacrarian development. And this message will make a little bit more sense in a minute when I show you one more thing. So far, so good, excellent. We have a better reconstruction of what the last common ancestor of deuterostomes looked like. But again, we were saying before, deuterostomes don't live alone isolated. Like the bigger picture also contemplates protostomes, because these are the two large groups of bilateria. And we were wondering if by comparing to other protostomes, we could also understand a little bit better the differences between the last common ancestor of deuterostomes and that of bilaterians. So for that, what we did was perform one-to-one comparisons, pairwise comparisons with Owinia fusiformis, which is early branching annelid that was thoroughly characterized at the genomic functional level very recently. And what we did was, yet again, a very similar analysis where we compared the transcriptomes of Ticodera during development to those of Owinia during development. And what we saw is that hemichordae gastrulation is more similar to the elongation stages of Owinia, having an enrichment in skeletal morphogenesis, neuron fate commitment, and anterior-posterior patterning. This is reminiscent of what I was discussing before about the similarities between hemichordate and cephalochordate developmental stages. And overall, this makes us think that deuterostome gastrulation mechanism predate the origin of this group, that the processes occurring after bilaterian gastrulation could have been deployed a little bit earlier in ambulacrarian development, and overall, that gastrulation is the deuterostome stage that has the highest molecular resemblance. This is a very nice big sentence that basically means that if we want to talk or think of any developmental stage that is important at, understanding the similarities between deuterostome, that should be gastrulation. So knowing this and knowing all of the things we figured out thanks to gene expression, then we try to understand what was the gene regulatory layer that was happening, genome regulatory layer that was happening there. And because as I said before, like many of these genes are shared between species, and it's not so much about what genes are there, but more like how are these regulated and how this regulation changes throughout the genome, right? Throughout evolution. So for that, what we did was we profiled the chromatin accessibility during the genome of the whole process of gastrulation, including also blastula. And we try to identify regions of open chromatin that are dynamically accessible throughout this process of gastrulation, right? And we detected this group of 6 clusters of open chromatin regions. These are just a subset, not all of them are there. And then the interesting thing here is trying to find sequences that can be in these regions of chromatin that can be potentially binding sites for these transcription factors, right? To see if this dynamically accessibility of the genome throughout development corresponds to also potential different activities of different transcription factors during development. So for that, what we did was a multiple enrichment analysis. And what we saw is that there was a differential enrichment of different classes of transcription factors of different motifs throughout development, right? And interestingly and reassuringly, these are some of the transcription factor classes that we also saw in the previous analysis when I was discussing, that the difference between early transcription factor classes and late transcription factor classes during development, which is nice. Another thing that we did was we investigated, the degree of agreement between the accessibility of the genome and of regions in the genome and the level of expression of the genes that were in the vicinity of those regions, right? And this is how we saw that there tends to be a positive agreement between chromatin being open and nearby genes being expressed. We suggest that, this cis-regulation, we are recapitulating here with this data, it tends to be activating, which is nice. And then another thing that we did was wonder if we categorize genes between being, let's say, being or not being that important for development at the gene regulatory level, what can we say about the chromatin? Are there any difference in the way these genes are regulated by chromatin depending on this category? And that we saw that indeed what we call genes that are transdevelopmental, because these are genes that are important for development and they are also involved in genome regulation in one way or another, they seem to be associated to a higher number of these regions for chromatin that are differentially accessible during development. And this is reassuring, and this is in line or is recapitulating former studies where we have seen that there tends to be like a complexification of the genome architecture of this type of genes because of their importance during development, obviously. And again, this is nice because it goes in agreement with what we know. So far, so good. Again, we have two modalities of data. We have gene expression and chromatin dynamics. And so far, we have seen that there seem to be like parallel agreements between the two. But the question remains that whether can we use this data in any way that helps us reconstruct what we call the gene regulatory networks, right? Because at the end of the day, these are the types of molecular entities that are driving, like the differences between one part of the embryo and another, one moment on the embryo another, and how things get done. So lucky for us, the field of gene regulatory network inference has already blossomed and is giving us a number of computational tools that are able to leverage a lot of this data. And such is the case of this tool that is called Anansi. This tool is able to leverage chromatin accessibility, gene expression, the proximity between genes and regions of open chromatin, and the enrichment of motifs in these regions to create a score of likelihood of interaction between the genes that we call transcription factors, that we know that they are transcription factors, and their target genes, right? Which can or cannot be also transcription factors. And by doing this for, like blastula and gastrulation, which are the late gastrula, which are the two stages that we saw that they were the most different, we were able to generate these networks of transcription factor and target genes, two of them. Which this is like a massive amount of data. And the best way to try to analyze this is coupling it with information that we already have from these kind of genes. Like, for example, we can leverage the centrality of the genes in the network, which can be a proxy of how important they can be, with, for example, being of a certain class of transcription factor or being involved in a certain biological process. If, for example, in our case, we have in situ hybridization data, we can have certain spatial information there, right, about where the genes are expressed. And in this case, this tool also allows, as you will see, to compute what are likely the transcription factors that are driving the differences between the structures of the two networks. So that might be also interesting into explaining the differences between the cis-regulatory landscapes of these two developmental stages. So the first thing that we did was leverage the information of transcription factors centrality. And what we interrogated basically is how central is every transcription factor in each of the two networks. And as you can see in this scatter plot, there are some transcription factors that I highlight in orange that they tend to be more central in blastula compared to gastrulation, or the ones that I label a bit more yellow that are in the y-axis, they tend to be more central in gastrulation compared to blastula. This means that there is a switch between the importance that is given to a transcription factor in one network or the other of each stage of development, and that is very nice. And this difference is even more apparent when you consider the subset of transcription factors that are also involved in genome regulation during development, as I said before. And where we can see that there is a slightly larger number of transcription factors that are transdevelopmental that get incorporated into gastrulation compared to blastula, which is interesting because at the end of the day, if we think about it in a way, blastula is more concerned about generating more cells, in increasing the proliferation of the cells and so on. And one other thing that we thought is, okay, which of the two genes which of the two networks might have the largest amount of genes that we know that are expressed later in development, right, during the larval stages and so on. And here, this is where we saw that the late gastrula network, even if these genes are deployed later, as in different larval stages and so on, these genes tend to be already pre-embedded in this network, which suggests that, at the very least, these genes, even if they are still not expressed, they are being prepped, let's say, right, at the molecular level, like at the genome regulation level, for whatever is going to happen later on. So that's cool. And then another thing that we could do is, as I said before, we can leverage positional information thanks to having in situ hybridization data. And we basically subsetted this gastrulation network in the different subcircuits according to the different germ layers. And we were able to see that this property of the networks of centrality seems to aggregate, to agree to associate with the level of expression or the ubiquity of expression of a given gene in the embryo. And this is certainly so for the different germ layers. So, for example, genes that have a higher centrality, they tend to be more highly expressed, and genes that have a lower centrality, they tend to be more circumscribed to certain structures within the embryo, which is a way of thinking about whenever you do a network like this and you are talking or you're studying development, this suggests that you can perhaps consider measures like network centrality as a proxy to try to understand where in the embryo or where in your system these genes might be. So another thing that we did, as I said before, is that if you leverage these two networks together with gene expression data, you can see what are the transcription factors that are likely driving the changes between the structure of these two networks. And when we compare the two networks in this sense, we were able to retrieve what Anansi calls the top influential transcription factors, right? And among them, we find a good number of well-known developmental regulators, like, for example, guscoid, OTP, some beta-CYP transcription factors. We also find FOXA, which will be important in a minute. And then this is, again, reassuring that some of these transcription factors are also part of the group of transcription factors that we discussed before, that they switch it a little bit, their expression during development across species, right? And then one of them, which is FOXA, it kept popping up in the different analyses. I just wanted to show here the network of the influence because I think it looks pretty cool. But yeah, FOXA is one of these genes that we detected here and there again and again. It was also one of the genes that was important into driving the similarities between the different developmental stages of the species that we were comparing. It's one of these transcription factors whose expression got a bit delayed or redeployed a little bit earlier in Ambulacraria. So that's interesting. And on top of it, we actually found out that we were able to retrieve FOXA together with some of partners that are important in different subcircuits for certain structures of deuterostome development. For example, we found FOXA together with its partners of the endomesoderm kernel, which is a network that was described already in sea urchin. But importantly and interestingly, we also find FOXA together with partners of the axial mesoderm kernel, which is a gene regulatory network that is important to specify the notochord in chordates. And this is a structure that does not exist in hemichordates. So this, even if it's in silico, but it poses the question or the hypothesis that maybe these cis-regulatory interactions, they were already present and predate the origin of these structures, which is something that has happened before in biology. Again, needs to be tested, but definitely is something very interesting. So then we wondered if this can be validated in any way, like in general, like this kind of data, and whether we can be which level of confidence or certainty we can have about the things that we are seeing in the network and with this chromatin data. And for this, FOXA was an ideal candidate. And so what we did was basically we took the cis-regulatory elements that are in the vicinity of FOXA in the genome of Ticodera, the ones that we characterized during our ataxic data analysis, and we fused these in different constructs to a reporter, to GFP, and then we injected this into sea urchin embryos because hemichordates are not that experimentally tractable, but definitely, like by doing this, we could also interrogate whether the cis-regulatory dynamics of FOXA in hemichordates is able to drive the endogenous expression pattern that one might expect in, like in a normal case of development in sea urchin. And indeed, that's more or less what we found. We found that we found gene expression, we found GFP expression in the germ layers that match and make sense, which is endodermal, a little bit of the ectodermal, a little bit of the endoderm. And basically, we found them at the right time in the right place. And the different dynamics of expression of GFP based on which contracts we were using, this allowed us to classify some of these cis-regulatory elements as repressors or activators, which is very nice. And then in general, so in this regard, we think that this validates the ataxic data, and definitely it makes us happy and excited to suggest things like, for example, this cis-regulatory machinery that we see in FOXA in hemichordates might be evolutionary conserved with the rest of deuterostomes, and it also makes us happy and excited to formulate the hypothesis that this might be the case for many other transcription factors, right? And now is the time to start validating all of this data. So the take-home messages so far are that hemichordate cis-regulatory genomics expand our understanding of deuterostome evolution, that gastrulation is the developmental stage of highest molecular resemblance among deuterostomes, that the conserved cis-regulatory mechanisms during deuterostome development date back to the last common ancestor of bilaterians, and now we have new hypotheses about the presence of cis-regulatory mechanisms that might predate the origin of some structures, like, for example, the axial mesoderm. And then with this, I want to acknowledge all of the people that were involved in this work. I want to give thanks to Juan, who was like an incredible, he supported this publication a lot, like this work a lot. I also want to thank my collaborators in Taiwan, in Academia Sinica, Jay Lin, Yixin Su, Skyu, also the people from the Van Heringen lab, because they were the ones developing Anansi, and it was very nice to interact with them. And in essence, all of the people who have been around for this time, all of the members of the laboratory of Scarmeta and Juan. And also I wanted to dedicate this work to the loving memory of Jose Luis, who would have been very, very happy to see this published, I think. And with that, thank you very much.
Wow. Thank you, Alberto. I mean, what a wealth of knowledge. I was, throughout your talk, I was thinking, each and every graph that you've shown us, which are all pretty, but for someone who is not familiar with the work you do, you might look at the graph and you cannot even imagine how much work has gone behind each and every graph. So kudos to you and to all your collaborators,
Thank you.
the teams involved. Very, very beautiful work. So, as I've mentioned to you before we started recording this talk, my questions will be mostly like generic and basic because I'm a little far from your field. But you touched upon a few terms that I'm particularly interested because I like thinking of evolution at the genome level, and I like thinking, looking at the genomes almost as if we are uh, dissecting morphological traits, right? So when you think about, old school evolution, if you will, people were looking at morphological traits, like beak size, for instance, or, body dimensions. And I think of the genome as the same way. And you mentioned things like genome complexification, right? And I was wondering whether you could expand a little bit on that, because my understanding is that, you've shown us that some transcriptional signatures, right, are shared in different developmental stages from different groups. And you also shown us, chromatin accessibility signatures, right? But I got a little lost in the actual term genome complexification and how that transpires from your work. Could you expand a little bit on?
Right. Yeah. So that comes from one of the analyses that we did, where we were basically using certain metrics to as a proxy for this, because when we talk about complexification, it's one of these forever questions in biology of what is complex, what is not complex, when do you put, and it's a matter of boundaries, right? Like where do you want to start calling one thing versus another? It's quite relative. Um, so in this case, we were thinking about in complexity in terms of how many cis-regulatory elements you have associated as a gene. Right? If you are a gene, then you have 1 versus 2 versus 3 versus 5 versus 10, and what can this say about or suggest about how you are regulated, right? And in this sense, it's been seen, like for example, in one of the previous projects that was done collaboratively in this lab, which was the Amphioxus genome project. They also saw something similar, which is like in the process of the origin of chordates and of vertebrates, there were several events of whole genome duplications. And you can definitely call that complexification because you have suddenly double the amount, right, of genomic content and everything. And and this allowed a level of certain level of versatility of how things get redeployed. Genes that suddenly they are twice the amount of the same gene family that is able to do the same things, genomically speaking, right? And in this sense, what they saw in that project was like there was this whole genome duplication came associated with a larger number of cis-regulatory elements associated to genes, especially to genes that are involved in development and so on. And in this sense, complexification is understood as an increase in the amount of things you can do, let's say, right? At least that's how I understand it. The amount of things that a gene can do, or like in how many different ways it can be deployed, or how many inputs it can have, and how nuanced this regulation can be. And of course, there are many layers influencing this, not only the presence of regions of accessible chromatin nearby, but definitely this is one of the layers. And at least there seems to be enough signal, at least I think, to state these kind of things, that indeed this seems to be the case. And it was reassuring to see something like this in hemichordates, because definitely they didn't go through the events of whole genome duplication the same. They didn't go through this in the way that the chordates did and the stem lineage that led to vertebrates did. But still, like, there seems to be this situation that genes that are involved in developmental processes, they tend to have like a higher, again, we're talking about trends, right? Like a higher degree of regulation of this level of input, and as I was discussing before, compared to other genes. And I wouldn't be surprised if this was also the case in, as people wrongly say sometimes, like, less evolved, or it's not less evolved, like other lineages in animals. Like, definitely, definitely.
Wonderful. And
I hope that answered your question a little
no,
bit.
absolutely. I must say that even though I don't work with developmental genetics and genomics, I think it's fascinating and it gives us an opportunity to look at these, large scale trends, right? Like across, phyla, if you will. And I think it makes us think a lot about evolution, which is what I love. Um, Building on what you mentioned, actually, um, I also wanted to return to a concept you touched upon, which is the centrality of
Mhm,
certain genes or transcription factors in a network. And
mhm.
you mentioned that that centrality actually has some predictive power, but I don't remember actually. So, the more central a gene is in a given network, what is the expectation that we would
Right,
have?
right. Yes. So, I mean, these will centrality is a measure that, I mean, first of all, I'm not like super expert or anything in graph and network analysis, but the way that I have understood centrality is a measure that it's intrinsically related to the way that you have constructed the network, right? And in this case, by extension, it's related to the type of data that this comes from. And for example, in this case, this association between that I show here in this slide now, this association between centrality and the ubiquity, I said, the way where it's expressed and how much is expressed anywhere, it can be, and it probably is somewhat linked to the type of data that we have, which is bulk expression. And in the end, because centrality is calculated in this network, sorry, I'm jumping around a lot, sorry, but centrality is calculated in this, as I said, it's part of the it's a byproduct of how these scores between genes are calculated, right? Which is basically a correlation of gene expression, level of gene expression, chromatin accessibility, motif enrichment, and so on. But one of the big components in this score is gene expression, right? And because we're talking about bulk data, which is basically whole embryos during development, etc. So genes that are present in a large amount of cells are going to be more expressed in the data compared to genes that are less expressed, because they are in a small group of cells. This is both a caveat and also, if you think about it, it's a caveat of the method because it definitely showcases that you can. It's a caveat and an exploit. You can think of it as, okay, you are recapitulating something that is intrinsic to the data, and the way you collected it. But at the same time, if you don't have this in situ information and you are characterizing new genes and you see that they are peripheral in the network, but still you can detect their expression, you can start thinking, okay, maybe that's because they are expressed at a certain place only. This is what I meant a little bit, right? When I am pretty confident that when you reconstruct networks like this, or you make networks like this or network analysis like this, but you don't start from this type of data. You are talking about single cell data. You account for like these differences in expression. Like this centrality will have a different meaning, and right. But again, caveat and blessing at the same time. At least how I see it.
Wonderful. And I do have one last question that during your entire talk, I was waiting for you to say something like co-option or exaptation of certain genes. But then I realized, so for instance, one of your take-home messages is that gastrulation is the developmental stage with the most molecular resemblance, if I'm not mistaken, right? Between
Mm-hmm, mm-hmm,
the groups you've compared.
mm-hmm.
And again, if I'm not mistaken, you've shown us that uh, you were able to show that certain genes are activated in slightly different developmental stages, right? So you have those transcription signatures coming in different moments of that development in different groups. So when I saw that, I first thought, well, these the transcription factors or the genes, or perhaps even let's say, the gene regulation apparatus is being co-opted or accepted to act on different moments. But then I also realized that if they're doing the same thing, but just in different times, it's not necessarily co-option, right? Because again, oh, just a second, there's someone knocking on my door. Just a second.
No, no problem. No worries.
I wasn't sure what it would be about. But anyways,
No
going
worries.
back to what I was saying, because to me, co-option or exaptation, which is similar, right, is when one trait or gene, you name it, whatever you're studying, is suddenly in a different group used to something
Mm-hmm.
very different, right?
Mm-hmm.
The classical example being feathers, right? So we believe that feathers, they were first fixed for thermal regulation, but now they are used for flight. Uh,
Mm-hmm.
so the genes and gene regulatory mechanisms you've shown us, they're not really changing their function. They're just being used at different times. So what would be then the evolutionary term for that? Because I think there is one. I'm vaguely thinking of, this change in timing or tempo, if you will, of the expression.
Mm-hmm.
What am I pointing to the right direction?
Right. Yeah. Yeah. I think you might mean heterochronies. Is
Yeah,
what some people.
heterochromeurate. Yes, thank you. Thank you. That's
I
what
think I think I think that's a little bit where some people like that work in this field use this terminology, like, and they talk about heterochronies, about how things get shifted temporally. This is also part of what has been done in the past in this field by by in the Amphioxus project, like the project that I was discussing of Winia fusiformis, this annelid, that was also characterized in a similar way to this. They discussed these things in terms of heterochronies. Basically, when something is being expressed and is happening, you infer that it's happening based on gene expression in one organism versus, or in one lineage versus another versus another versus another. And this is how I didn't show here, but like some other analysis that we did were a little bit different in the way that this comparative analysis, in the way that we conceive it, which is instead of doing this pairwise transcriptomic divergence, with these heat maps that I showed, basically what you do is you infer these groups of temporal expressed genes, like these big clusters of genes that, it's like these waves during development. You compare them pairwise. So basically you how many genes from the same gene families are in one time of development according to what one species is telling you versus another, basically in which clusters, in which timing of development they are expressing this species or in that species. And this is how they define heterochronies. And I think it was in the case of zebrafish and Amphioxus when they were comparing, like Athelios, a fish with a vertebrate, with Amphioxus, they saw this kind of patterns. And definitely they also saw them in this annelid functional genomics paper. And we also have this kind of analysis in the paper. And you can see agreements, and again, which you could call homochronies, and then these disagreements, which you would call heterochronies. Um, I kind of agree with you that I don't know, at least at the very least, just by looking at when they are expressed, we should be cautious about calling them co-option or not, because we still need to disentangle where they are expressed. If, for example, such is the case of this project, right, which is just the temporal dimension. Aside from this very same slide that I am at, where we have in situs and so on, like, but the rest of it is mostly like the temporal dimension only. It's also about data. This is also one of the reasons why I think it's very exciting. People are jumping into single cell technologies now, to single cell transcriptomics and so on, to try to do these projects and this kind of visions of and designs, but including also like this cellular resolution that somehow you can also map to somewhere in the organism if you have the right data. And then I guess that's when you can start asking yourself, okay, if I see this gene or this group of genes that I see in one species in this time and in this group of cells, and I see it in another group of cells, what is going on, right? And then, but of course, like the question becomes more complicated, because then you start talking about cell fate and the cell identity. And then it's a new layer, which is the evolution of cell identities and cell types, which is something very, very, very interesting, because as far as I understand, again, no expert, and correct me if I'm wrong, or feel free to add up on this, but cell types, it's only now that they are being considered evolutionary substrates or Darwinian entities, something that can change. And this is something very, very interesting. I remember when I was writing the PhD, one of the things that I was writing about during, like the origin of animal multicellularity and like multicellularity in itself, why does this trait emerges, and how it gets fixed, and what is the reason behind, etc. And it's basically, it opens a new window for selection to take place, right? And I don't know, I find it very nice. But definitely coming back, sorry, that was a digression, but coming back to the question, yes, co-option right now, I'm not sure, but heterochrony, I think it's a safe word, let's say. And definitely it keeps you on track of what you're doing. And the good thing is that with single cell now, all of these studies are going to get even more exciting, more complex, but more exciting. But yeah,
Mueller, right? Well, this is beautiful. Yeah, I agree with you about the terms, and I think one of the very beautiful things, or perhaps I think one of the conclusions that, at least to me, it stands out is that evolution can do a lot with little, right? I mean, little relatively, meaning that you don't need to have the emergence of many new different gene families or even different genes, with the same families of genes, with the same, handful of transcription factors, you can tweak things and have, very different species and body plans and things like that. So I think this is a message that I mean, I guess anyone in biology uh, could see that these days reading the literature. So yeah, well, thank you. I must say, yeah, your
right.
work is very, very interesting, very beautiful. Kudos to you, to the team. You've presented a wealth of data and knowledge. And
Thank you.
yeah, and I think I do not need to interrogate you anymore. So,
No, I'm excited to discuss about these things. To be honest, I was having memories, good memories about when I did my PhD Viva. It was a long discussion. And it was full of things. I was very exciting. So don't
yeah.
worry about that.
Yeah, the philosophical aspects of the work in biology, I think sometimes are, the most fascinating parts, because it's
Yeah,
so easy to get, bogged down into the, trying to parse the data, trying to get the right graph and trying to, treat data and find the right way to present stuff. But then we often
mm,
forget the big picture. And when we remember it, it's very nice.
right.
But anyway, I will stop the recording now. And then your talk will be coming up in the coming weeks. Okay. So thanks again
Perfect.
for teaching us so much today. I'm very
Thank
happy
you.
to have you over here today. Okay. I'll stop the
Thank you.
recording.
Thank you, Mateus. Thank you.